Welcome students. Today we will discuss nucleic acids, nucleotides, nitrogenous bases, tautomers of bases, nucleotide derivatives and nucleotides as regulating molecules. The objectives of our today's lecture are to understand nucleic acids, nucleotides, nitrogenous bases, tautomers of bases, nucleotide derivatives, nucleotides as regulating molecules. Let's begin with nucleic acids. Nucleic acids are very long, thread-like polymers made up of a linear array of monomers called nucleotides. There are two kinds of nucleic acids: deoxyribonucleic acids or DNA and ribonucleic acids or RNA. DNA is found mainly in chromatin of the cell nucleus, whereas most of the RNA, that is 90%, is present in cell cytoplasm and a little, that is 10%, in the nucleolus. The nucleic acids are the hereditary determinants of living organisms. Now let's discuss nucleotides. Nucleic acids are biopolymers of high molecular weight with a nucleotide as their repeating units. Nucleotides are the phosphate esters of nucleosides, and these are components of both RNA and DNA. RNA is made up of ribonucleotides, whereas the monomers of DNA are 2'-deoxyribonucleotides. As regards to their elemental composition, the nucleic acids contain carbon, hydrogen, oxygen, nitrogen, and enough phosphorus. In the ribose moiety of ribonucleoside, C1 and C4 are involved in furanose ring formation, while in the deoxyribose component of a 2'-deoxyribonucleoside, C1 and C4 are involved in the furanose ring and C2 does not bear a hydroxyl group. Upon hydrolysis, under different set of conditions, the two nucleic acids yield three components: phosphoric acid, a pentose sugar, and a nitrogenous base. Each of the three components of nucleic acids are: first is phosphoric acid. The molecular formula of phosphoric acid is H3PO4. It contains three monovalent hydroxyl groups and a divalent oxygen atom, all linked to the pentavalent phosphorus atom. Second is pentose sugar. The two types of nucleic acids are distinguished primarily on the basis of 5-carbon keto sugar or pentose which they possess. One possesses D2-deoxyribose, hence the name deoxyribose nucleic acid, while the other contains D-ribose, hence the name ribose nucleic acid. Both these sugars in nucleic acids are present in the furanose form and are of beta configuration. An important property of the pentoses is their capacity to form esters with phosphoric acid. In this reaction, the OH groups of the pentose, especially those at C3 and C5, are involved, forming a 3'-5' phosphodiester bond between adjacent pentose residues. This bond, in fact, is an integral part of the structure of nucleic acids. Now coming to nitrogenous bases. Two types of nitrogenous bases are found in all nucleic acids. The base is linked to the sugar moiety by the same carbon, that is C1, used in sugar-sugar bonds. The nitrogenous bases are derivatives of pyrimidine and purine. Owing to their pi electron clouds, both the pyrimidine and purine bases are planar molecules. Now we will understand nucleotide derivatives. First are pyrimidine derivatives. These are all derived from their parent heterocyclic compound pyrimidine, which contains a 6-membered ring with 2 nitrogen atoms and 3 double bonds. The common pyrimidine derivatives found in nucleic acids are uracil, thymine and cytosine. Uracil, which is found in RNA molecules only, is a white crystalline pyrimidine base with 112.10 Daltons and a melting point of 338 degrees Celsius. Thymine found in DNA molecules only has molecular weight equal to 126.13 Daltons. It was first isolated from thymus, hence so named. Cytosine is found in both RNA and DNA. It is a white crystalline substance with a molecular weight of 111.12 Daltons and a melting point of 320 to 325 degrees Celsius. Now coming to purine derivatives. Purine derivatives are all derived from their parent compound purine, which contains a 6-membered pyrimidine ring fused to the 5-membered imidazole ring and is related to uric acid. The prevalent purine derivatives found in nucleic acids are adenine and guanine. Adenine is found both in RNA and DNA. It is a white crystalline purine base with a molecular weight of 135.15 Daltons and a melting point of 360 to 365 degrees Celsius. Guanine is found both in RNA and DNA. It is a colorless insoluble crystalline substance with molecular weight of 151.15 Daltons. It was first isolated from guano bird manure, hence so named. Other purine derivatives are also found in plants. For example, caffeine 1,3,7 trimethylxanthine is present in coffee and tea, and theobromine 3,7 dimethylxanthine is found in tea and cocoa. Both caffeine and theobromine have important pharmacological properties. Now coming to physical properties of nucleosides and nucleotides. Owing to their polyionic character, nucleic acids are soluble in water up to about 1% weight by volume according to size and are precipitated by the addition of alcohol. Their solutions are quite viscous and long nucleic acid molecules are easily sheared by stirring. Ionization. The acid-base behavior of nucleotide is its most important physical characteristic. It determines its charge, its tautomeric structure, and thus its ability to donate and accept hydrogen bonds, which is the key feature of the base-base recognition. Hydrogen bonding. The mutual recognition of A by T and of C by G uses hydrogen bonds to establish fidelity of DNA transcription and translation. The NH groups of the bases are good hydrogen bond donors, while the sp2 hybridized electron pairs on the oxygen of the base CO groups and on the ring nitrogens are much better hydrogen bond acceptors than are the oxygens of either phosphate or the pentose. This base pairing pattern rapidly became known as Watson-Crick pairing. There are 2 hydrogen bonds in an AT pair and 3 in CG pair. While Watson-Crick base pairing is the dominant pattern, other pairings have been suggested, of which the most significant to have been identified so far are Hookstein pairs and Crick-Woobel pairs. Francis Crick proposed the existence of Woobel base pairing to explain degeneracy of the genetic code. This phenomenon calls for a single base in the 5' anticodon position of tRNA to be able to recognize either of the pyrimidines or alternatively either of the purines as its 3' codon base partner. Now coming to spectroscopic properties. Neither the pentose nor the phosphate components of nucleotides show any significant UV absorption above 230 nanometer. In such ordered structures, the bases can stack face to face and thus share electron interactions that profoundly affect the transition dipoles of the bases. Typically, such changes are manifest in a marked reduction in the intensity of UV absorption by up to 30%, which is known as hypochromicity. This phenomenon is reversed on unstacking of the bases. This phenomenon is used in determination of temperature-dependent and pH-dependent changes in base stacking. Secondly, it permits the monitoring of changes in the asymmetric environment of the bases by circular dichroism, that is CD, or by optical rotator dispersion, that is ORD effects. Now coming to shapes of nucleotides. Nucleotides have rather compact shapes with several interactions between non-bonded atoms. We can more simply describe the shapes of nucleotides in terms of 4 parameters: the sugar pucker, the syn anticonformation of the glycosidic bond and the orientation of C4-C5 and the shape of phosphate ester bonds. Now we will understand tautomerism in nitrogenous bases. Compounds that exist in two structural isomeric forms which are mutually interconvertible and exist in dynamic equilibrium are called tautomers, and the phenomenon is termed as tautomerism. Tautomers are obtained through the redistribution of protons within the molecular system. Thus, tautomerism does not cause change in chemical formula, but the structure of the molecule is changed due to movement of proton from one site to another. The 4 bases of DNA can exist in at least 2 tautomeric forms. Adenine and cytosine can exist in either amino or amino forms, and guanine and thymine and uracil, which are cyclic amides, can exist in either lactam, that is keto, or lactam, that is enol forms. The tautomeric forms of each base exist in equilibrium, but the amino and lactam tautomers are more stable and therefore predominate under the conditions found inside most cells. A tautomeric equilibrium involves alternative structures that differ only in location of hydrogen atoms. The keto or lactam form predominates at neutral and acid pH values which are of physiological importance. The enol or lactam form becomes more prominent as pH decreases, that is at acidic values. Next is nucleotide derivatives. The 5 nitrogenous bases were once believed to account for total base composition of animal and plant nucleic acids and were hence designated as major nitrogenous bases. It is now known that other minor bases called modified nitrogenous bases also occur in polynucleotide structures. The 5'-methylcytosine is a common component of plant and animal DNA. In fact, about 25% of cytosine residues of plant genome are methylated. Similarly, 5'-hydroxylmethylcytosine and its glucoside derivative and pseudouracil. Methylation of purines, particularly of adenine, in DNA is now known to occur in the genetic material of microorganisms, and it is believed that plant genomes will also be shown to have methylated purines. The 6-methyladenine is found in bacterial DNA. Next is nucleotides as regulating molecules. In addition to their roles as subunits of nucleic acids, nucleotides perform some other functions. First is nucleoside analogues as drugs. Recently, 2 nucleoside analogues, 3'-azidodeoxythymidine, that is AZT, and 2'-3'-dideoxycytidine, that is DDC, have been therapeutically used for the treatment of acquired immune deficiency syndrome, that is AIDS patients. Secondly, as carriers of chemical energy. Nucleotides may have 1, 2 or 3 phosphate groups covalently linked at 5'-OH of ribose. These are referred to as nucleoside mono, di and triphosphates and abbreviated as NMPs, NDPs and NTPs respectively. The 3 phosphate groups are generally labeled as alpha, beta and gamma starting from the ribose. NTPs are used as a source of chemical energy to drive many biochemical reactions. Adenosine triphosphate or ATP is by far the most widely used. When coupled to a reaction with a positive free energy change, ATP hydrolysis shifts the equilibrium of the overall process to favor product formation. Next is as components of enzyme factors. Many enzyme cofactors and coenzymes such as coenzyme A, NAD, and FAD contain adenosine as part of their structure. They differ from each other except for the presence of adenosine. Thirdly, as chemical messengers. The cells respond to their environment by taking cues from hormones or other chemical signals in the surrounding medium. The interaction of these chemical signals with receptors on the cell surface often leads to formation of second messengers inside the cell, which in turn lead to adaptive changes inside the cell. Often, the second messenger is a nucleotide. One of the most common second messenger is nucleotide adenosine 3-5- cyclic monophosphate, also known as cyclic AMP. Cyclic AMP serves regulatory functions in virtually every cell outside the plant world. Guanosine 3-5- cyclic monophosphate or cyclic GMP occurs in many cells and also has regulatory functions. So concluding here, nucleic acids, DNA and RNA are biomolecules made from monomers known as nucleotides. Nucleotides are the phosphate esters of nucleosides, and these are components of both RNA and DNA. Each nucleotide consists of three components: a 5-carbon sugar, a phosphate group, and a nitrogenous base. RNA is made up of ribonucleotides, whereas the monomers of DNA are 2-deoxyribonucleotides. Two types of nitrogenous bases are found in all nucleic acids. The nitrogenous bases are derivatives of pyrimidine and purine. The common pyrimidine derivatives found in nucleic acids are uracil, thymine, and cytosine. The prevalent purine derivatives found in nucleic acids are adenine and guanine. The oxygen-containing nitrogenous bases exist in ketoenol or lactam forms. The tautomer containing the carbonyl group, that is CO, is designated as the keto or lactam form, and the other one having hydroxyl group, that is OH, attached to doubly bonded carbon is referred to as enol or lactam form. However, it is the keto or the lactam form which predominates at the neutral or acid pH values, which are of physiological importance. It is now known that many other minor bases called modified nitrogenous bases occur in polynucleotide structures. In addition to their roles as the subunits of nucleic acids, nucleotides perform some other functions. Recently, two nucleoside analogues, 3-azidodeoxythymidine or AZT and 2-3-dideoxycytidine, that is DDC, have been therapeutically used for the treatment of acquired immune deficiency syndrome patients. Nucleotides may have 1, 2 or 3 phosphate groups covalently linked at 5-OH of ribose. These are referred to as nucleosides. They are used as source of chemical energy to drive many biochemical reactions. Adenosine triphosphate or ATP is by far the most widely used. Many enzyme cofactors and coenzymes such as coenzyme A, NAD and FAD contain adenosine as part of their structure. So students, with this we conclude our today's lecture. Hope you have understood it well. Thank you.